Okay, we're going to talk about crossing over here. So I'm going to try and explain crossing over. And of course, we have pairs of chromosomes. So let's say these are what we call homologous chromosomes. And that means that they essentially are the same. They have the same sets of genes in the same locations. So the locations are called loci or a locus. So let's say this particular gene is located here. That means on the homologous chromosome, say this is a chromosome 1, you have two of this particular gene. Let's just call this one A, for lack of a better term for it. So this is gene A, and it's located at this particular location on let's just make it up. Let's say this is chromosome 7. So chromosome 7, and we have gene A that's located here. Now there may be other chromosomes, of course, other genes on this chromosome. So this one might be B. That's located here. And let's put at least one more. Let's put, let's see, C isn't good because you can't tell capital from small. So let's go gene E here. And you'll see that one over here. Now these are supposed to be the same size. I'm just not very good at drawing on this map. So on this program, so chromosome 7 has 3 genes, and of course each organism, especially if you're human, I should say, not each organism, but each human organism, you're going to typically have 2 chromosomes that are going to be homologous, and homologous simply means, of course, that they have the same genes at the same loci or locus, or roughly the same locus. And what happens with crossing over is that when the organism is replicating the DNA, you make extra copies of these chromosomes right before the cell splits. And so let's see if I can draw this. This is kind of tricky. So let's say this one is a copy of that one, and this one is a copy of this one. Ew, that doesn't look good, but I think you get the idea. So these are duplicated during replication, and actually all 4 of these chromosomes are combined into what's called a tetrad. Now I'm going to draw them as little lines here, and I'm going to show that they're kind of wound around each other. So there are 4 of them. All right, and they're kind of wrapped around like ropes. Like you would do twine or something, and they get wrapped up like this. So you have 4 chromosomes, and now they're homologous. This one is going to be homologous to this one, and there are going to be little lines, right here. Here's where those genes are. Same over here. You're going to have the genes in the same spot, and they're all going to be wrapped around each other, though. And so you're still going to have like A up here and B somewhere in here and E down here, and they're going to be on all 4, and they're all going to get wrapped up. And what happens is when they get wrapped up like this, and they're called a tetrad. There are a couple of other terms that you might see that refer to the same thing, but the idea is this intertwined mass of chromosomes. There are pieces of a chromosome. So let's just draw a generic chromosome down here that are inherently weaker, shall we say, than others. So I'm going to do this like a dashed line, like perforation on a paper, so that it's easy to cut out. And there are going to be, there are going to be regions that are going to, let's just say, tear, for lack of a technical term here, easier than other regions. And what happens whenever you have this formation here with all 4 of them all around together, they do tear, and the little regions interchange with one another. So maybe let's say a little one here. Let's say this piece tears off, and what it will do is with one of the others, in that same region, they do have to be homologous. They have to be similar in the sequence to do this. But this same region here, they will exchange with one another. All right? Kind of like this. This piece goes over here, and this piece goes over here. Well, this is fine. They're the same chromosome. Why does it matter? Well, it matters because when you inherit the chromosomes from your parents, realize up here, if you inherited this original one from mom, all right, and this one from dad, there are differences between those. Our standard Punnett square, remember, mom and dad, you know, you may have big A, little a, and this one may be all big A. Well, if you do look at it this way, then these chromosomes, you might have inherited big A here, but you might have inherited a little a over here. All right? And you, instead of big B, you might have had a little b over here. And E, you might have, maybe these are all little e's over here. Well, if you notice these chromosomes, you never have if this were the case, if it were just like I have it written here, you never have a big A on the same chromosome as a little b or a little e. Or you never have a big B with a little a or a little e, or a big E with a little a, B and a little a, and vice versa on the other. You never have the combinations. In other words, there's always big A, big B, and big E, or little a, little b on the chromosome. You never have a chromosome that is, you know, like this one, which would be big A, little b, and say big E. This is not the way you normally think of genetics and the way you do Punnett squares. This shouldn't happen, all right? But it does. And it occurs infrequently, but it occurs often enough that you have to take it into account. And what is going on here is that crossing over is occurring. And we are going to use crossing over as a method to map things. But to understand how the mapping units work, you have to understand that this process is going on occasionally inside the genome. And what's important with this, and why this can be bad or good, is that maybe some of these genes go together. In other words, maybe there is some significance to always getting big A with big B and little a with little b. And I will give you an example of why that's important. Because let's say the A gene, that means big A or little a, involves your height. All right, so how tall you are. Tallness, tall, and we'll make this one your short. Oops, it's hard to write on this thing. So little a means you're short, big A means you're tall. It has to do with your height. And then let's make B have to do with your foot size. So here you have big feet and you have small feet. Well, this is interesting. Let's say this makes you, if you get big A, you have really long feet, like you wear size 14 shoes or something. And if you have little a, you wear size 4 shoes. Now big A, little a, maybe you're in between. But the idea is the same goes with the, I'm sorry, I was talking B there with foot size, so it's the size shoes. So you have really big shoes or really small, and maybe it's in between when you have the heterozygote. But up here, you know, up here you are like 6'4" if you have big A, and you're 5 foot tall if you have little a, and you're 5'8" or 5'9" maybe if you are big A, little a. Well, that would be fine, and there's nothing wrong with being different heights here, and there's nothing wrong with different shoe sizes. But when you combine them, they could be a bad thing. So, for instance, if I were to be big A, and be, little b, I would be 6'4", but I have these tiny little feet. You probably are going to have foot problems. Actually, you probably aren't going to make it. You probably have all kinds of other bone deformities because these genes that control the size of your feet and your height control many other things. And to tell you the truth, this child probably wouldn't even make it to birth. The bones would be two different sizes and everything wouldn't fit together. So it would be, especially if it didn't even allow you to be born, if the child died while in the womb, if they got this combination, then you know that this isn't going to show up very often in nature, and it's a survival trait in the sense, or I should say, it's a lethal trait, is what you call it, if you get this combination. If you get this combination, it's lethal to the organism, maybe, and they don't mature. And of course, you can imagine, what if you were 5 foot tall and had size 14 feet? You would be clumsy. You would fall over yourself. And so even if you survived to being born, you'd be clumsy, and the animal, if it's not a human, wouldn't be able to hunt or move around very easily. If it was something that was hunted itself, it might not be able to get away and be killed very quickly in life. So you would see that these combinations aren't going to probably be passed on very often, and they die off. Well, the result of this is that gradually what happens is traits that aren't supposed to be occurring at the same time, there are some mechanisms in how our chromosomes are designed to ensure that this doesn't happen very often. And what happens is that over time, there is a survival trait or a survival mechanism for genes to move closer together over evolutionary time if they are related to one another and need to be a certain gene, a certain allele, I should say, that needs to go with a certain allele of another gene. So, for instance, here, just a second. Okay. So, for instance, I have up here, these two need to go together. And so over time, what you will find, if genes need to go together and you can't have different alleles, you will find that they move closer together on the chromosome. All right. Little b, or I should say, let me make that little a and little b. And they will be very close to one another. All right, so A and B will slowly migrate closer to one another if they are bad for you, if the alleles are different and might be dangerous to go together. So in other words, if big A is dangerous or lethal if little b is associated with it, and vice versa, little a is dangerous with big B, then slowly these two will migrate closer to one another, and they will migrate so that they are within one of these little perforations. All right, and so what you would find is that in nature, if something is like this, like foot size and height that need to go together, you will find that these genes will be within the same little perforation that we're talking about. I'm leaving out the technical terms here for you. No need to really know them at this point. But you will find that A and B are close to one another, and therefore will be here. The others, if it's not important, then they can be anywhere. All right, so E could be up here. No problem if it has nothing, maybe it's eye color. Eye color has nothing to do with the processes of you standing up and moving around and agility, so therefore you don't have to worry about it, and E could be anywhere. But A and B will have to be close and will have to be within the same little region. And so you should never get crossing over with A and B if they are related in some way, where if the alleles, like the two dominant alleles here, big A and big B, if they are related in such a way as that they need to stay together, they must both be dominant, or and on the other one, the two recessive ones must stay recessive and must stay together. If that's the case, then they will migrate closer to one another over evolutionary time. We're talking about millions of years here. But over time, they will move within these perforations, and then you don't have the problem with crossing over. So I wanted to explain this to you and explain how there is a distance between these, and how some genes are really close together, and they need to be, and other genes are further apart, and they're not related to this because they're not related in this type of relationship like we had with big feet and height.